Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about the introduction of cells, tissues, and microscopy. We want to preview cell ultrastructure, or the details in the cells. We want to preview cells, which collectively together make tissues, and tissues collect together make organs, and then organs get together organ systems. We want to have an overview of light and electron microscopy, and a little bit about preparing specimens to be able to see at light and electron microscopy, and a little bit about section orientation. Now, in terms of looking at histology, there's a breakdown of things. The smallest living substance is protoplasm, and protoplasm resides in a cell. A cell is the smallest unit of protoplasm. Some animals only have a single cell. So a cell is the simplest, smallest unit of life, and protoplasm is the living substance that's inside there. If you get several cells that do the same type thing, that makes a tissue, and there's four basic types of tissue. There's muscle, nerve, connective tissue, and epithelium. If you get more than a couple types of tissue, you get an organ. Organ functions as a unit: skin, kidney, intestines, blood vessels. And if you have several organs, you get an organ system: respiratory system, digestive system, reproductive system, the immune system, different systems. So we start with a cell, the smallest unit of life, and both animals and plants are composed of cells, tissue, organs, and then organ systems. If we look at a cell, we see a cell here. This is an animal cell. And it shows you the cell membrane around it. It goes to the nucleus and the cytoplasm therein. If we look in there, we can see right above the nucleus would be a Golgi apparatus, which looks like a series of pancakes with vesicles coming and leaving the Golgi apparatus. They're coming from the endoplasmic reticulum. So this is the rough endoplasmic reticulum. It's rough because it has ribosomes, and also it's rough ER because it has like little stacked sacs, as opposed to smooth endoplasmic reticulum, which is a series of anastomosing tubes. And then we have the nuclear membrane itself, the nuclear envelope that surrounds it, with nuclear pores for things for the messenger RNA to get out of the nucleus. We have a nucleolus inside there, which is responsible for making the ribosomes. And then we have the mitochondria for energy. We have a lipid, lysosomes inside there. We have centrioles and secretory granules are some of the things that we see in the cell. So there's a cell, and then a tissue is a group of cells doing the same type, like epithelium. Or this is epithelial cells as a tissue. And then if you get more than one tissue, we have connective tissue in through here. We got nerves, we got epithelium, then that would be an organ. And then the organ system is where you get more than one organ together. Here we get the digestive system. See this is the liver, and here is the gallbladder inside there. So it was in 1660 that Robert Hooke looked through a primitive microscope at a thin cut piece of cork. So he was looking at cork, and he saw a series of wall boxes that reminded him of tiny rooms, or cella, occupied by monks. Monks lived in little tiny rooms named cella, and so Hooke coined the word cell for the little boxes that the nuclei and cytoplasm are in. He saw that in a cork. And so here we can see cells in plants in a cork. It would be, you see the little boxes that the cells are in, more boxes. There's a nucleus, nucleolus, and the cytoplasm, and actually we're seeing the cell wall. Although animals don't have cell walls, they are composed of cells. So the cells are the building blocks of life for both plants and animals. And that brought us to the cell theory. All organisms are made up of one or more cells. Okay, cells are the basic building block to life. And then the third thing, all cells come from existing cells. So these two gentlemen added to what Hooke saw as cells, and then they added to the observation about characteristics of the cells, and that brought us to the cell theory. All things are made up of one or more cells. Cells are a basic unit of structure of life. All cells come from pre-existing cells. Cells contain hereditary information, which will pass on to the daughter cells. So during mitosis, you want the cells to divide to produce a cell just like the parent cell. All cells are basically the same in chemical composition. They have hydrogen, oxygen, nitrogen, different components, sulfur, different components in there, and basically all the cells are the same. Just the ratios of these is what makes them different. Now there's 4 basic types of tissue. We have epithelium, and epithelium sits on the basement membrane that we can see right there, and this one has an amplified brush border as well. There's the nucleus, and so this is one cell. There'll be another cell over here. There's also connective tissue. We see the nuclei of fibroblasts in through there, nuclei of fat cells in through there. Muscle tissue, see nuclei of muscle cells, striations, big striations here, which are the intercalated disc of skeleton of cardiac muscle. And then we see nerve, and this is a nerve cell body. Here, there's a nucleus and a nucleolus in the center, and the little nuclei there are the satellite cells that support it. So if we look at epithelium, that's one of those types of cells. It covers organs. So on the outside of your kidney or liver or the lungs, you would have that, and also lining viscera, lining the cavities, lining blood vessels, and the secretory cells of glands. All those are epithelium. So here we see the cardiovascular system. You would have epithelial cells lining the entire surface of it. A distinguishing feature is that all the cells sit on the basement membrane. So this is, we see one cell here, there's a nucleolus, and then this is a cytoplasm of a cell, and there's a basement membrane. And also, if you have one cell, a thick layer, we call that simple. If you have multiple layers, we call it striated. And another characteristic of epithelium is that they are attached to one another with these junctions. So you have tight junctions, a different type of junctions, hold cells together, and as a consequence of them being held together, they can make a lumen, or a cavity is what they have. So if we look at simple squamous, squamous being flat, simple being one layer, as opposed to stratified squamous, again, the flattened cells are on the very surface. You always name it by the cells on the surface. So these look the same as those, but there's multiple layers here. And so this is stratified squamous or simple squamous. This is simple cuboidal, little cubes, as we see, and simple columnar. So these are columns. If you have more cytoplasm above the nucleus than you have between nuclei, then that will make it columnar as opposed to a cuboidal cell. There's also pseudostratified columnar, and those are cells that all the cells touch the base, they don't all touch the top. It looks like it's stratified, but it's really not. And then there's transitional, with its big cells on the surface that can lay open and the cytoplasm unfolds, the cell membrane unfolds, I should say, as the cell stretches, and that's transitional, and that's associated with the urinary tract. Epithelium can have specializations. It can have amplified surface. There's microvilli amplifying the surface there. Also, it can have cilia. And here we see ciliated cells in through there. Again, we can see the amplified surface that has a PAS staining. It's sugar, stains for sugar. So all these are rich in sugars. There's the nucleus and the basement membrane that we can see. So we'll have some of these specimens from the Body Worlds. We want to thank and acknowledge Body World for their contribution to the images in here. Here we see one. This is an individual. See the muscles in through there, and see his skin has been removed. So if we look at skin, we can see an epidermis out here and a dermis below. So epithelium and connective tissue down below. Connective tissue is vascularized and it provides nutrients for epithelium that's avascular. Connective tissue is a histologic glue. It binds things together. These are the fibroblastic nuclei that we can see. That in between there we see a lot of space in through there. Those are the collagen fibers that help connect things together. So there's connective tissue. Of the connective tissue, there's different types. What's important in connective tissue is the extracellular matrix. So we name it for the extracellular matrix. Loose connective tissue is few fibers and lots of cells. A dense connective tissue is a lot of fibers, few cells. And cartilage is a avascular type of connective tissue, and that is at the end of bones. And it's also the skeleton of you as you were early in the embryo. And then there's bone. Bone is calcified matrix that we see. So all these are connective tissue. Here we see some nuclei of fibroblastic cells. They're kind of like cigar shape. But all this pink in through here is really collagen, extracellular matrix. Here we see a fat cell. There's another fat cell there. Here's this nucleus right in through there, another nucleus right in through there. So this is connective tissue. Cartilage, as we see here, bone is connective tissue too. So we can see the bone and we can see the back fat. As we can see, there's thicker on the individual that's 300 pounds than one that's 120 pounds. So if we look at fat here, we can see muscle. And then there's muscle that we can see in through there as well. Muscle, the function of muscle is to generate contractile force, is what it's doing. So it has a high concentration of contractile proteins, actin and myosin, and they can either be arranged diffusely as in smooth muscle, or they can actually form sarcomeres, organized layers of fibers, and you see those in skeletal muscle and cardiac muscle. And here we can see the striations that are brought about by having these cells line up, as you see. So we have skeletal muscle, which is a big cell. Nuclei are on the sides, on the periphery. Cardiac muscle, branch cell, one or two nuclei in the cells, and it's branched so that it can join to other cells. Smooth muscle cells, as we see here, is kind of cigar shape, and we can see them here and here as well. So smooth muscle, we have smooth muscle in through here, which are lined like cigar-shaped cells, as we see. And then cardiac muscle with intercalated disc, and then skeletal muscle with the nuclei on the periphery and the striated muscle. So here we can see where the body whorls had this guy. The muscles have walked out of the skeleton that we can see. So we see muscle in here, and here is nerve. This is nervous tissue that we see in here. And the function of nervous tissue is to transmit, receive, and integrate electrical signals. That's what it does. So neurons are very large excitable cells that possess axons and dendrites. And these could be quite long. Could be a meter length. And there's different types. There's bipolar, pseudo-bipolar, pseudo-unipolar, and then multipolar, unipolar. And so here we see a bipolar one. This one is in the bipolar cells in the inner ear. So this is a cell body with a pole going in either direction. There's also glia cells, which are support cells. Now, a nerve is a collection of neuronal processes brought together by connective tissue. So on the outside in through there, we have connective tissue. And sometimes the axons, this was gray, here's axon, gray, and there's an axon. It can be myelinated. And so it has a myelin sheath around it, or in other cases, it can be unmyelinated. Here we see a myelin sheath around this one. Here we see some axon here of unmyelinated axons as well. And so a nerve, we can see the axons here and here, and the myelin sheath that surrounds it with connective tissue in through there. We call that connective tissue endoneurium as opposed to a perineurium. That would be a heavier band that we see. So you have those in a central nervous system. You also have peripheral nerves. And here we see a peripheral nerve. If you look at these are satellite cell bodies. This is a nissal substance. The granularity that we see is really ribosomes and rough endoplasmic reticulum ribosomes. And then we can see the big nucleus, very euchromatic light staining nucleus, and a big nucleolus is what we're able to see. In most organs, you will have some peripheral nerves located to have the final control over the process that organ would have. And here we see nerves. Nervous tissue can run from your brain all the way down to your spinal cord, from your spinal cord down to your legs. And so that is a nerve. Another connective tissue is blood cells. And the function of blood cells is to carry oxygen or carry CO2. And the white blood cells are transient inhabitants of blood. They're produced in the bone marrow, and they're to help be defenders, a part of the immune system. And then there's platelets, and we see the platelets here. So these are red blood cells, white blood cells, and then these are platelets, and platelets is for clotting the blood. So the red blood cell, biconcave, that's what we see in here with a smaller thing. White blood cells, usually lobulated nucleus with some granules. And then we have platelets that has no nucleus, but it has similar cellular functions of the cell, and they're produced by the megakaryocytes in the bone marrow. So here we see different cells. These are the eosinophils, basophils, neutrophils, and through here, it does have granules, but not as big as these red ones and blue ones. And then the monocytes, you can write to macrophage, and then lymphocytes, as we see up there. Lymphoidal tissue, you would encounter that whenever you're looking at tissues. You may just have infiltration of lymphocytes. But there's amplification of the immune response with lymphokines. And so the lymphokines are produced. This is a T cell helper cell that's producing interleukin-2 that's going to stimulate other helper cells and also the T killer cells. And then they also have influenced the B cells, which are producing plasma cells to produce antibodies. To lymphoidal tissue, here we can see a lymphoidal. These are Pryor's patches in the gut. These are intestinal villi that we have here. And so you'll have some along the airway and along your digestive tract because you're being exposed to the outside environment. And you will find a high concentration of them in the thymus, spleen, lymph node, and then these Pryor's patches that you see. Now, endothelial cells are special epithelial cells, and that's what lines blood vessels. So we have one here, we have some here. There's an endothelial cell nucleus right there with red blood cells inside there. They're also line this lymphatic. So both lymph and blood vessels have endothelium, which is a special epithelium derived from the mesoderm. And then if we look at blood vessels, there's all kinds. They're artery or arterial, capillary, as we see there and there, the smallest one, and then the venules, and then also the lymphatic brings lymph back to the bloodstream. So we can see the many blood vessels. Here's a spermatic cord. There's an artery, very well defined. Here's a vein, and we see a valve in a vein, another valve in a vein in through there as well. And there would be capillaries. These little guys in through here would be the capillaries that we can see. Now, if we look at the basic types of tissue, we see epithelium is the epidermis. Also epithelium in the eye. Epithelium in the gland. Here's the salivary gland. Epithelium on the tongue. Connective tissue would be the dermis. Underneath the epidermis, it'll be bone. It'll be the white fascia or gray fascia that surrounds muscle, that kind of tough whenever you're eating your muscle. Also, teeth are embedded in connective tissue as well. Muscle, we can see muscle here. There's muscle that moves the eye. There's muscle that moves the hair. If you've ever seen a cat or a dog with his hair standing up on his back, it's called a muscle contraction, pulling that hair up. Also, the tongue has muscle in it for you to be able to move. Nervous tissue in the eye, in the brain. Inside of most organs, you have ganglion cells, as we talked about, for nerve cells. And then, of course, you got taste buds on your tongue. If we look at a microscope, our eyeball is up here, ocular lens, prism, and then you have the condenser lens down here, objective up through there. Light comes through, and you're able to visualize the light that goes through the specimen. Here we see a scanning electron microscopy view of the biconcaveness of the red blood cells. So these are red blood cells, and then these others are the white blood cells. So magnification and resolution. Magnification just increases the size of things, but you can't just keep increasing size because you don't gain any more information. What's important is resolution. It's the smallest distance that can be distinguished between 2 points. If you ever look down the railroad track, as you look down, it looks like the railroad track components are getting closer, but you know they're not. If they were, the train wouldn't work. What you're doing is losing resolution as you look down the track, and so no longer can this distance be distinguished. Only that distance can be distinguished at the other end. So magnification increases size. Resolution actually has a calculated formula. You have a constant, which is 0.161, times the wavelength of light or wavelength of electrons divided by the numeric aperture. And here you can see in light microscopy, we can see micrometers, but with electron microscopy, we can see nanometers. So we have log difference in the magnification between the two. In terms of preparing specimens, we first of all have to fix it. You fix it to prevent it from decaying, is what you do. Remember, whenever they thawed up Iceman, he started to decay. So they had to freeze him again quickly, because when they thawed him up, he was not fixed. He was just frozen. And then he started to undergo degradation, which is what would happen all along if he hadn't been frozen all these years. And then we embed it in paraffin plastic to make it hard. If you cut a hard-boiled egg, you can pretty nicely cut. But if it's not hard-boiled, then you can't cut it so nicely. So in tissue, you want it firm, like a hard-boiled egg, or harder. And that's why you put it in these plastic or paraffin. Fix it, plastic, and then you have to section it. So you section either micrometers or nanometers for the different types. So you fix it, you dehydrate it, you clear it with benzene, you embed it with paraffin or plastic, and then you make these sections in it. And here you can see how you make the sections. You put the piece of tissue on this motorized extension, and that just goes up and down. And as it goes up and down, it advances half a micrometer or so. And then as it does, that part is cut off, and you take those and make a microscope slide once you've stained. Here you can see where they are making sections of the brain in through there. And then you have to stain it. So with light microscopy, we use H&E. H&E is hematoxin eosin. Hematoxin stains nuclei. We can see some nucleolus there staining very nicely. And the eosin stains protein red, which is part of the cytoplasm. And then toluidine blue stains everything. And you have to look at the size, shape, and density there to see it. And so there's PAS stain, staining for carbohydrates. And you can see the basement membrane is carbohydrate rich of epithelium. And also in this case, the brush border of the proximal tubule is PAS positive, as well as the basal lamina inside the glomerulus itself, toluidine blue. So H&E, PAS, you can go by the color codes for these, but not with toluidine blue. Shape, size, intensity of stain. So there's the H&E, electron microscopy. You have electron opaque stain, so you can stop electrons from coming through. osmium lead citrate you might use. And then there's fundic stomach that we can see. This is H&E. These are intestinal, this is the pits, the gastric pits inside the stomach. And here we can see in the stomach surface mucus neck cells, and then this is chief and parietal cells that are in there. Chief and parietal cells, this is the surface mucus cells that we can see in there. Here we see H&E and toluidine blue of the eye. So there is rods and combs in the eye. Here we see intestinal absorptive cells. You can see it's simple one-layer column cell, simple columnar. We can either see junctions between adjacent cells through there. We can see mitochondria in through there as well. And there's the nucleus and the nucleolus and the brush border, and this would be where the basement membrane would be. And the cells are attached by junctions, and you can see those junctions right there and there. These are junctions that allow these cells to connect to one another so they form a lumen or a cavity. We can see those cells again. There's the brush border. These are goblet cells that produce mucus inside the intestine. Now, the reason you stain tissue is because tissue is largely transparent. We're looking through our cornea right now, looking through the lens because it's transparent. But if you stain it, it changes the amplitude, so you're able to see the difference in what's stained and the background. As opposed to unstained tissue, it changes the phase, but it doesn't change the amplitude. So a regular light microscopy with unstained specimen looks like that, so you can't see. In fact, it looks like this, where the stain did not penetrate very well. But when you stain it, it differentially stains, so it shows you some things, dark nuclei and lighter lumen that you can see. The phase contrast microscope lets you observe living cells, where bright field microscopy, which is what this is, the cells are all dead. But here you can see, you can look at the mitotic activity of a cell. So bright field microscopy is what we usually use with stained specimens, and this is one that's not stained, so it shows you it needs to be stained. But phase contrast, you can work with living cells. These are dead, these could be living. You can also see them with differential interference contrast microscopy and dark field. So these are the ones that let you see live cells. Bright field, what we use most of the time, are all dead cells. Here's an electron microscope. You sit right there, look at a fluorescent screen through these ocular lens, and there's your specimen up through there, your condensers that you have involved. So we have a light source, which are electrons, and the electrons come through. You have a magnetic lens that changes the focus the light on the electrons on the specimen, and then from there you project your focus to focus on the fluorescent screen. And here we can see electron microscopy versus the light microscopy. You can see it much better here because the resolution is better, because this uses electrons and this uses white light. And since electrons are much smaller, a log smaller than the wavelength of white light, you can see a lot more detail with electron microscopy than you can with light microscopy. Here you see a sperm head with a lot of membranes, or nuclear membrane, interarchitectural membrane, outer architectural membrane, and the plasma membrane. Here we see scanning electron microscopy of a domestic pen, like you pin your clothes with. You see it looks sharp there. It doesn't look that sharp right there. And also there's other structures on there. That is bacteria. So there's bacteria on there. So when you stick something in you, you're actually inoculating yourself with the bacteria. Here we see some cells showing its pseudopodia and spikes there as it interacts with the environment. So if we look at light microscopy, the light source is light, white light, and it has glass lenses, and you view it with the eye. In contrast, if you use electron microscopy, you have magnetic lens and electrons, and you view it on a fluorescent screen. With scanning electron microscopy, you have electron magnet, electron source, but it's the second electrons and not the first one. It's the second one that is going to expose to a detector, and the detector will feed that back on some kind of cathode ray tube. So you see it so different places. You can see it with the eye or film, you can see it with a fluorescent screen, or you can see it on a television tube. There's other light and electron microscopic procedures. There's immunofluorescence, and here we can see where the actin filaments, clusters thereof, are located in the cells. These are stress fibers of the cell, and so that was detected where actin is by, you have antibodies, and hence immunofluorescence, antibodies that's bound to a fluorescent tag that's unique for actin. Then autoradiography is where if you would use radioactive sugar in a cake, then the cake would be radioactive. So in this case, you feed it, the cell, something radioactive, precursors, and then it's incorporated in the protein. In situ hybridization is where you use some of the code from the DNA to look for sites that can bind to that specific code. Freeze fracture is kind of like freeze drying, and you can fracture the cells, and etching is freeze drying, I should say. So if we have antibody here against an antigen, a protein, and it's fluorescent tagged, then we're able to see where that antigen is, where that protein is, indirectly by looking at the tag. And so this is really for light microscopy. For electron microscopy, you need something electron opaque, and so peroxidase makes a product that's opaque, electron opaque, and then also gold. So you can have gold of different sizes attached to the antibody, and the gold will show you where that antigen of interest is. You can amplify it by having antibodies against antibodies. Autoradiography is where you fed the cell some radioactivity precursor, and then it's incorporated in its final product. SCS geoelectrophoresis is a way that we look at proteins in membranes. If you use SDS, it will dissolve the membrane, as you can see here, and make the protein soluble, where they can migrate in a gel. And when they migrate in a gel, a bigger protein migrates less. Smaller proteins migrate more, as you can see, and we use that compared to a standard to tell us what kind of molecular weight we have. Here we have Kamasi blue staining the cytoplasm. There's a nucleus staining the cytoskeletal components that are in there. Autoradiography procedures to localize the proteins within a gel or a cell by the fact that it is self-radioactive. So if the cell is radioactive, like this is a rough VR, then you put a photographic emulsion on top of that, and wherever the electrons are coming through, they will expose the film, and that's how you're able to do it. So the autoradiography is a direct measure of film by the beta alpha particles, or gamma particles. The fluorography is exposure of a film by secondary light. So the primary radioactivity hits something and makes a secondary light that you actually see on the fluorescent screen. So you shine the light on the fluorescent screen, and that fluorescent screen gives off light, and that's what you're seeing. So in situ hybridization, you take a little piece of DNA, and then you're able to look into the chromosomes or wherever to find where that little piece of DNA would be located on the intact chromosome and which chromosome it will be associated with. Photomicrographs, as we can see, are from light microscopic level. This is one from light microscopy. And the dark staining here is showing you where the DNA corresponds. For example, you can find, if you have a probe
For bacterial DNA, you can find where the DNA or whether or not you would have the same type of sequence of DNA in your cells, and I'll tell you, you do. Carbon replica is now we're looking at the replica. This is the replica, not the specimen. There's a specimen, and so you fly in platinum or gold or something, and it piles up on this side, kind of like a mountain. It rains on this side, not that side. So it piles up. Then you put carbon on it to support it, and then you pull that off, and you can see the difference in this, and that will tell you what things look like. So this is regular conventional electron microscopy, and this is carbon replica electron microscopy. You can see the nuclear pores, you can see the organelles, see the plasma membrane. So in terms of section orientation, if you had a water hose and you cut it like this, it would be a cross section like a donut. Cut it back this, looks like railroad tracks. Cut it over here, a solid track. And then here, again, like a donut or oval, all depends upon, and we see all those in these nerve axons. So these are the axons in here and the myelin sheath around it. And so we can see those that are nice cross sections. We can see those that are like parallel, like a railroad track. We can see big heavy sheets like this, where there's only one. And then you can see oval and round ones below. So if we look at intestinal villi, basically they have projections that project out into the lumen. But if you cut right straight across here, it'll look like this. Looks like something different. But really, all these are attached down there like that one. Now, in terms of orientation, if you section through this, you can see if you section through there, it'd be a single cell thickness. If you section through there, it looks, you're looking through the tops of some, through the nuclei of others, and through the base of others. And so it looks like there's multiple. And here, along here, if you section through there, it looks like there's a bunch of cells. But it just looks that way, but it's not. And this, what you want to remember, is a planar section. That's what this is, whether it's like this or that. Can make it look like there's more layers, which looks like more than one, but it can never make it look less than the actual number. So when you section through there, if it's one, it's going to look one. If it's two, it's going to look two. At some place in there, it's going to look two. You can never make it look less. It can only look like there's more. Again, orientation, where you cut through the top, you don't even have a lumen, or you cut in through here, or you cut through the middle where you have exposed the individual. So remember, planar section can make it look more, never make it look less. I hope that shined a little bit of light on electron microscopy and light microscopy. Here we see more. That's just a dawn rising, the light as we are able to see there. Next time we're going to talk about membranes and receptors, and we want to thank the books which were the original source. I did not generate these images. Some of them I did, but as a rule, they got from textbooks who had excellent examples of illustrations of things, and those are the original sources. I want to acknowledge their contribution to this. If this presentation was useful, please share it with your colleagues and your friends so they can use it to enhance their knowledge of histology. Thank you.